Welcome back. Let's talk about some white blood cells. So white blood cells I'm going to abbreviate as WBCs. White blood cells are named white blood cells because they don't stain red whenever they're stained, and red blood cells tend to carry that red pigment, so that's why white have been given their name white. But whenever they're actually stained with a couple other things, they actually look pink and purple, which makes them not white, which is very, very frustrating whenever you're first learning this for the first time. So they're white blood cells, they're not red blood cells, they're different than red blood cells, and they go through a different process and they become different cells, different types of cells than red blood cells entirely. The reason why we talk about them in the blood chapter is because they are a part of that Buffy coat. They are a part of that 45% of the formed elements, where they are a very important cell that is traveling within the bloodstream in order to get to the target tissues, in order to help the immune system respond as it needs to. So for this chapter, we're just going to talk about briefly the names and generally what they do, but in the immune system, we're actually going to talk about how they all talk to each other. All right, let's go ahead and get to it. So white blood cells have another name. So just like red blood cells are named erythrocytes, white blood cells are going to be also named leukocytes. So leuko referring to the as part of the lymphatic system. White blood cells have nuclei and they do have organelles. Compared to red blood cells do not have a nucleus, but they do have some organelles, but they do not have a nucleus. White blood cells aren't carrying hemoglobin. That's not their primary response. They're not carrying hemoglobin. And we definitely don't have as many of them as red blood cells. So here we have 5,000 to 10,000 white blood cells for every 4.2 to 6.3 million red blood cells. They travel through the vascular highway of the cardiovascular system in order to get to the tissues, and they use chemical messengers to know where they need to go. So if there is a cut or a tear or an inflamed tissue somewhere, that tissue is actually going to send chemical signals to go and activate those white blood cells or recruit those white blood cells to that area, and then the white blood cells are going to do what they need to do to clear the infection or the inflammation or whatever they need to do as they see fit. All white blood cells can migrate out of the bloodstream. So this means that at any time, if you have a blood vessel like this, it can actually pierce its way or push its way through the endothelium lining of the red blood of the blood vessel in order to actually get to the tissues that are surrounding those blood vessels. We call this margination, immigration, and diapedesis. So margination is when they are starting to poke their way through the red blood vessel, or through the blood vessel, excuse me. Immigration is when they actually pass through the blood vessel, and then diapedesis is when the blood vessel starts to close itself off so nothing else leaves, and that the cell actually gets to the tissue itself. White blood cells are always attracted by a chemical stimuli, so they're always following a positive chemotaxis. Chemo means chemicals, taxis means movement, so we have a chemical movement. We're always following a positive chemical movement in order to actually get to whatever tissue it is that we're supposed to get to. If you ever take an advanced immunology course or a pharmacogenetics course or a cellular biology course, you might go into the details of this stuff, and so that's why we introduce it here. We have two different types of white blood cells. We have granular cells and we have agranular cells. Anytime we have the prefix A at the beginning, that means without. So the granular cells have granules. The agranular cells do not have granules. So we have granular leukocytes and agranular leukocytes. The granular sites include neutrophils, eosinophils, and basophils. The agranular sites include monocytes and lymphocytes. How did we know they were one or the other? We stained them a couple times. One stained a little differently than the other, and so we separated the two between the two, or we separated them in those two groups. So the granulocytes means they have little granules within their cell. So if I were to look at the cell and they had the little nucleus here, it might have granules in the center of it, and those granules actually stained a certain color. Versus an agranulocyte would have the nucleus here and no granules in the cytoplasm, and so nothing is there to stain. All types of lymphatic defenses are almost non-specific, except for some of these cells. These cells are actually, they vary between not specific to very specific. So we'll talk about their specificities as we go through this. And then lymphocytes are responsible for specific defenses, like we'll talk more in detail about whenever we get to chapter 22. All right, so all of the cells as white blood cells, there's 5 of them. There's neutrophils, eosinophils, basophils, monocytes, and lymphocytes. All right, if you want to have a good way of remembering all 5 of those, there's an acronym or saying, never let monkeys eat bananas. So never for neutrophils, let for leukocytes, monkeys for monocytes, eat for eosinophils, and bananas for basophils. If that helps you remember them, awesome. But do whatever seems fit for you, and I'm sure there's other naughty ones on the internet if you're interested. Let's go through these one at a time. So first is neutrophil. Excuse me. Okay, so a neutrophil, we have, neutrophils are probably the most abundant type of white blood cell we actually have in the bloodstream. Accounts for 50 to 70% of the white blood cells. They're a granulocyte, and their granules are relatively neutral, and so they actually don't, let me make sure. Yeah. So the granules don't actually stain, which makes them a little difficult to call them a granulocyte, but they are there, so that's why we call them a granulocyte. They're really dense, and they usually have multiple nuclei. So neutrophils, multiple nuclei, NN. Neutrophils, multiple nuclei. And multiple nuclei means that there are multiple lobes. So as you can see in this nice picture, it's not just one nice spherical nucleus. It looks like there's maybe 4 or 5 different nuclei in there that are all kind of sandwiched together, kind of maybe sharing a same membrane. Usually neutrophils are one of the first ones at the site of injury. So if there's an injury, the chemical mediators usually send neutrophils over first. And neutrophils, whenever they get there, they will ingest and digest the bacteria or the pathogen, and then as a result they will explode, and the explosion involves pus. So acne on your face is basically a bunch of dead neutrophils that ate a bunch of bacteria on your face and then exploded and then turned into little pockets or divots in your face, and that's acne. Fun. Super fun. The way they work, just like I mentioned, they will find the bacterium or the pathogen and they will eat it, and then they will destroy it, and they will also have like a self-destruct method, so part of that will turn into pus. Okay, next is eosinophils. Eosinophils are named eosinophils because they stain red with eosin dye. So eosinophils stain red with eosin dye. Eosin is a red dye. Everyone knows that, of course. Ha ha ha. I'm rolling my eyes. Big exaggeration. How do I remember this, Alexandra? Well, eosinophils are going to stain red, and eosinophils are very important in attacking and killing parasites. They will do a very good job of surrounding the parasite and flagging it for the rest of the immune system to come and destroy it. What else helps kill parasites? Well, the antibody IgE, or immunoglobulin E. And that's how I remember eosinophils. E are attacking parasites, just like IgE would also attack parasites, and we'll talk about IgE towards chapter 22. But that's how I remember that eosinophils are attacking parasites, and they're red for eosin. It's a relatively smaller number of cells. It's not the smallest, but it's definitely not the greatest number of white blood cells we actually have. The way they kill off the parasite is once they surround the parasite, then they release their granulocytes, and those granulocytes are super toxic to the parasite, and they basically shock the parasite to death. So, terrible death. Okay, next is basophil. Basophils are the smallest percentage of our total white blood cell count. They're named basophils because they're stained with a blue dye, and that blue dye is named something with baso. I forget what it's called. I totally forget what it's called. But so B for blue. You're going to look at them and go, oh, Alexandra, it's not actually blue. It's kind of purple. You lied to me. I think it looks blue sometimes. Sometimes I think it looks purple. So, but the eosinophils definitely look red or pink. So definitely make sure you identify the difference between the two. Basophils are also going to stain very darkly, and it's going to be a difficult time trying to differentiate the nucleus from the rest of the cytoplasm, because they're also going to have a bunch of granulocytes or granules within the cytoplasm that will also be released and release histamine, which will produce inflammation for any of the nearby tissue targets or parasites nearby. Basophils blue, eosinophil red, eosinophils parasites, basophils histamine. Next is monocytes. Monocytes, mono means one, cyte means cell, so this is a one cell. and they are very big eaters, so big cell, big cell. They're going to have a massive nucleus, so big nucleus, maybe a lot of cytoplasm, but much bigger nucleus than any other cell you're going to see in the scope. It might be a bilobe nucleus, it might be a pretty spherical nucleus, but it's going to be massive. It'll be big. These monocytes eventually turn into macrophages. Macrophages mean big eater, macro big, phage eater, so big eater. And they will also help recruit other neutrophils to the site whenever they're attacking their infected cell of choice. These are in the second to last greatest abundance of cells that we have for the white blood cells. Last but not least, we have the lymphocytes. The lymphocytes are going to account for the second largest majority of the cells. Lymphocytes, lympho referring to the lymphatic system, cyte referring to cell. And these are named in reference to lymphocytes or to the lymphatic system because they will turn into one of three specialized lymphatic cells, either a B cell or a T cell or a natural killer cell. We will talk way more in detail about what the T and the B cells do in chapter 22. But for now, B cells will talk to T cells in order to learn what to do and how to kill the pathogen. And natural killer cells, they're natural killers. They will target things that need to be killed, and they will kill them. Similar to a neutrophil, except they don't create pus on their way out. A lymphocyte is going to have a relatively big nucleus, not as big as a monocyte, and the nucleus to cytoplasm ratio is going to be a much bigger difference than a monocyte. A lymphocyte is going to have a big nucleus, small cytoplasm, versus a monocyte might have a big nucleus and also quite a bit of cytoplasm. This makes sense because a lymphocyte is going to have a lot more genetic material in order to actually differentiate itself into a B or T or natural killer cell, versus a monocyte is going to eventually turn into macrophage, which means it needs to have a lot of space in order to eat stuff, so we're going to need a lot of cytoplasm to do so. Okay, some terminology: leukopenia, leukocytosis, and leukemia. Leukopenia, so leuko referring to leukocytes, penia referring to lack thereof, so we have an inadequate number of white blood cells. Leukocytosis, so leuko referring to leukocytes, or the lymphatic system and leukocytes. Cyto referring to cell, cis means making of, so, cytosis is making of those cells, so an excessive number of white blood cells. Leukemia is the the extra side of that, so kind of like the tumor form, leukocytosis, where we have way too much and there shouldn't be that much. If we're actually talking about how we create these white blood cells, we are talking about leukopoiesis, and that's the production of white blood cells. Again, we're starting with the same hematopoietic stem cell, and we're either turning into a myeloid stem cell to become a granulocyte or a monocyte, or we're turning into a lymphatic stem or a lymphoid stem cell where we're turning into a lymphocyte. I highlighted the boxes in red of what's important to understand from that graphic, so hopefully that'll make sense to you by now. A lot of information about how we actually make these white blood cells is unknown. So we show you this flowchart, but this flowchart is actually different in a lot of different textbooks, and it's constantly being changed. So there's a lot of researchers that are like, no, monocytes come from, myeloblast cells, and we actually have dendritic cells that can change and turn into monocytes, or they can turn into neutrophils just by willy-nilly. No, not just by willy-nilly. There's a lot of science that actually goes into all of that. But this flowchart is relatively standard and accepted by most folks. Just understand that if you go into an advanced course, they might actually say like, oh, well, we actually understand that these cells come from this cell lineage, and so on and so forth. So, but for the most part, this is what's generally accepted by, by a majority of folks. Okay. How do we regulate this? Like I said, a lot is unknown. Whenever we're making those those lymphocytes, we're making the pre-B and pre-T cells. Those pre-B cells become mature B cells in the bone marrow, and those pre-T cells, they originate from the bone marrow, but then they actually become mature in the thymus. Eventually, the B and the T cells are actually going to be matured once they're exposed to certain types of antigens. And then sometimes some of the cells will only be activated whenever they're in the presence of other cells. So this is all like chemical signaling, cellular biology to the T. There's just so much stuff going on with how we actually produce these cells that we just don't have enough time to talk about in this course. But if you're ever interested, definitely check out the book that I'll suggest during the lymphatic system chapter, or take an advanced cellular biology course. Okay. Up next is platelets, and we'll chat about that in the next video.